200 years after the publication of the well-known Frankenstein novel, research teams worldwide have not only come as far as to create hybrid creatures such as human-rat chimeras in the laboratory, but also have come up with concrete plans to intervene in evolution for the first time. We will give you the most interesting examples on these Frankenstein-like projects and on the concept of genetic nuclear bombs. Welcome to Clixoom Science Fiction.
1, 2, 3.
For some time now, genetic engineering teams do no longer shy away from experiments on humans. And it is really sensational how far biomedicine has come so far. At the same time, research in this area is also scary and gives me goosebumps more often than not. Take this as an example. In China, scientists have created genetically modified embryos, and they could have actually grown them into people. Yet, that did not happen. The embryos were not delivered, but that's probably just a matter of time. Specifically, they inserted intact gene segments into genetically diseased human embryos. In 3 out of 6 cases, the correction of the genes was indeed successful. All this brings us to a time when reproductive medicine will be quite normal in designing people. The research in neurobiology is heading in a bit of a different direction. Scientists are creating so-called brain organoids. Human stem cells are differentiated into brain cells. These then actually grow into a mini brain. It's hard to believe, but the cells make connections and form 6 cell layers typical for the cerebral cortex. Could these brains then begin to feel or think? The participating research teams dismiss these doubts. The mini brains would indeed have no sensorial input, and also their size is way too small. So far, the mini brains are not supplied with blood. As soon as they are as big as a lens, the supply of oxygen and nutrients is no longer sufficient. But that's exactly what a neurosurgeon from the University of Pennsylvania wants to change. Oh man, his experiments are really incredible. His research team is fusing these almost mini brains with the visual cortex of rats, and with success. So these tiny artificial brains are being supplied with blood and sensory impressions. And yes, you can really put it this way. These human rat chimeras make it possible for human tissue to look through rat eyes and thus perceive the world. I have a really bad feeling about this. How big is the danger that these creatures created in the lab will turn against us at some point? Frankenstein sends his regards. Actually, there are more and more news from this area. Biomedicine and genetics take over engineering tasks and manipulate life as it pleases. The key to this is the so-called CRISPR technique. This gene cutter makes the manipulation of genetic material very precise and is also very inexpensive. In addition, the method is very easy to use, even for beginners. In the near future, organ donors may become superfluous because the plan is to breed human organs such as kidneys in pigs, for example. Other research teams aim to revive extinct species such as the mammoth. Another very successful area of research is synthetic biology, which is used to design bacteria. A few projects that are already in the process of being implemented: bacteria that log how their host's disease progresses, or bacteria that can see in 3 colors. Finally, let's look at a project that would really be revolutionary. In Burkina Faso, a research team will very probably intervene directly in evolution for the first time soon. The specific plan is that in the farming village of Bana, mosquitoes will be released that have an infertility gene in their genetic material. According to the computer simulations carried out so far, this gene will then spread to the entire mosquito population. The idea of the research team working on behalf of the Bill and Melinda Gates Foundation to stem malaria. Because the malaria pathogen uses these mosquitoes as a host, and these will probably only live on in a reduced number. If the plan works, then man might actually be able to outsmart nature. Incidentally, this method is referred to as gene drive in science language. Critics call it a genetic atom bomb, because the genes will probably spread as quickly as it is the case with a nuclear chain reaction. All of these projects can somehow be associated with the Frankenstein novel, but how much have we learned from this? Will today's experiments in the gene laboratories of this world end in a disaster for mankind? One thing is certain: you will not be able to stop these experiments, but we need authorities that control such projects concerning genetically modified creatures and then examine whether they benefit humanity. Now, if you're asking what will science try to cure next, check out our videos on nanobots. Click here. Scientists are working on fixing our bodies from the inside. Remember to subscribe and don't forget the bell. Stay tuned and see you soon.